In this lesson, we'll be focusing on viruses, their structure, and the life cycle. It's really important when we talk about viruses to separate them from what we've already talked about, which was cells. We spent a lesson talking about prokaryotic cells, which we essentially focused on bacterial cells, and then eukaryotic cells, which humans are an example of an organism that has eukaryotic cells. But being microbiology, we tend to focus on eukaryotic cells as it relates to microorganisms, things like fungi and protozoa. With viruses are not considered cells, so they are off in their own little category, so we want to separate them from the way we think about typical cells. So, if viruses aren't cells, what are they? They are what we define as being obligate intracellular parasites. What that means is they must infect a living cell, and once they get inside of that living cell, they essentially hijack the cell into reproducing and manipulate the living cell into producing more viruses for it. So they are dependent on living cells in order for survival. And we typically think of viruses as these infectious agents that target human cells, and certainly that's true when we're thinking about it from a more healthcare lens. But there are many different types of viruses that target all different types of cells. So there are viruses that target bacteria. We identify those as being bacteriophages. But there are viruses that target plants, viruses that target fungi, and yes, there are many different types of viruses that target animal cells such as human cells. It is important to understand that a virus can only replicate if it's inside of a living host. So if an individual virus is just sitting on a surface of an inanimate object like a pen or pencil or doorknob or desk, it is only ever going to be one virus. A virus, in order to produce more of itself or replicate itself, must infect a living cell, and then once it gets into the living cell, it essentially hijacks the living cell's machinery and manipulates the cell into producing more viruses for it. But it's not able to do this independently without the help of a living cell. And viruses are not considered cells. They don't have any of the typical structures that a cell would have. For example, eukaryotic cells, such as our own, have things like a nucleus and many different other membrane-bound organelles like Golgi apparatus and endoplasmic reticulum and mitochondria. And I understand that when we talked about viruses, we said that they didn't have many of those structures either. Or when we talked about bacteria, but bacteria at least have ribosomes that they can use to make proteins. Viruses don't even have that. So with viruses, they essentially are just some genetic material surrounded in protective coverings that, and because they lack all of their kind of metabolic machinery that's typically needed for things like replication, transcription, and translation, because they lack all of that machinery, they're not able to replicate on their own. So their whole goal is to infect a living cell and then hijack the living cell's metabolic machinery and manipulate the cell into producing more viruses for them. So there is a debate within the scientific community as to whether we should classify viruses as being living or non-living organisms. So some scientists believe that they should be classified as non-living because in biology, biogenesis, which is basically defined as living organisms produce other living organisms of the same kind. So humans produce humans, cats produce cats, dogs produce dogs. That's kind of how we define a living organism based on its ability to produce more of itself. And with viruses, technically, they are not able to produce more of themselves independently on their own. In order for them to produce more of themselves, they have to first infect a living cell. So if they do not infect a living cell, they're not able to produce more of themselves on their own. So some scientists believe that we should classify them as non-living for that reason. Other scientists believe we should classify them as living organisms because while they're not able to replicate independently, they still know how to invade a host cell. And once they invade the host cell, they know how to take the host cell over and manipulate it into ultimately producing more viruses for it. So sometimes scientists believe that they should be classified as living due to that ability to be able to kind of manipulate processes within a cell. For whatever reason, it's never been definitively stated whether they're living or non-living, so we tend to refer to viruses as either being active or inactive. So if a virus is active, that means it still is able to infect a cell. If the virus is inactive, it is no longer able to infect a cell. Many viruses have different time periods that they can be active for on a surface. So, for example, some viruses are active on a surface for only a few hours, whereas some could be several days to potentially several weeks. And as long as the virus is still on a surface and within that period of being active, it can infect a host cell and then ultimately lead to something like an infection. However, if it is past that time limit of activity, if it gets into a non-active state, then it no longer is able to infect a host cell and cause an infection. So we know that viruses are not cells, but what makes them so unique and why aren't we able to classify them as being cells? One of the primary reasons is that both prokaryotic and eukaryotic cells have DNA and RNA. So both, for example, human cells and bacterial cells have DNA as their genetic material. That DNA can be transcribed into RNA, and then that RNA can be eventually go through translation to form a protein. So both types of cells, whether it's prokaryotic or eukaryotic, both cells have DNA and RNA. With viruses, it's always one or the other, never both. There are some viruses, such as the influenza virus, that are RNA viruses, and they will have RNA as their genetic material. And then there are some viruses that are DNA viruses, and they will have DNA as their genetic material. But at this point in time, there's no viruses that have both DNA and RNA. So it's always one or the other, but never both. Whereas cells have both. They have DNA and RNA. And the other distinct difference is kind of what we've already been talking about. Again, cells can grow and divide. Our cells are able to produce, well, most of our cells are able to produce additional cells. Bacterial cells are able to produce more bacterial cells through a process known as binary fission. So cells are able to produce more of themselves. Again, viruses are not able to do that independently. Again, a virus must infect a cell and then essentially hijack the cell's metabolic machinery and manipulate that cell into producing more viruses. So viruses are, again, highly dependent on the living cells that they infect. I do want you to understand that viruses are very small. There's not a specific size range that I need you to memorize, but I just want you to understand they're much smaller than what you're probably used to. So this image kind of puts that into perspective. Some things that you may see in other courses looking under the microscope would be things like white blood cells and red blood cells if you went through the anatomy courses. So these are obviously human cells, and we can kind of see where they are on the size chart here. We have bacteria down here. It doesn't say what type of bacteria, but possibly something like E. coli. And we can see how small the bacteria are compared to something like a red blood cell. And then viruses are much smaller than that. So we have the coronavirus here. We have a bacteriophage, which is a virus that can infect bacteria. And then we have something down here known as the Zika virus, which is a viral infection that we'll talk about later on the semester that's transferred through mosquitoes. But I just want you to understand, viruses are the smallest of the microorganism. I mean, viruses are so small that one of our respiratory cells could hold approximately 1 million influenza viruses. So if you think about how small an individual cell is in our body, and the fact that that cell could hold 1 million viruses, it will kind of put it into perspective at how small viruses are. But viruses are the smallest of the microorganisms that we'll talk about in this course. So viral structure is very simple because viruses don't have a lot of components, which is why they're so dependent on living cells, because they don't have any of the machinery that's typically required for things like replication. So going through their structure is very simple. You definitely should know the standard parts of a virus. So all viruses would have what we call a nucleic acid core, which is what we're looking at here in the center. The nucleic acid core is where the genetic material is held. If you remember from other courses, there's two different types of nucleic acid. There's DNA and RNA. So with viruses, their nucleic acid core is essentially where you're going to find their genetic material, which is either DNA or RNA. If it's a DNA virus, it'll be DNA. If it's an RNA virus, it will be RNA. And then surrounding the nucleic acid core as a means of protection, you have this purple layer here called the capsid. So the capsid always surrounds the nucleic acid core, and it is meant to be a protective layer. All viruses have a capsid that surrounds their nucleic acid core and serves to protect their genetic material, which is really the most important thing a virus has. Its genetic material tells it how to infect a host cell and then ultimately how to take over that host cell once it gets inside. Now, some viruses, that's all they have is their nucleic acid core and then a protective capsid to surround it. If that's the case, then that virus is referred to as a naked virus. Naked viruses only have one protective layer known as a capsid. There is another class of viruses called envelope viruses, which is kind of what we're looking at here. Envelope viruses have an additional layer, so they have a capsid, but then they have an additional layer of protection known as the envelope that surrounds the capsid. So that's what we're looking at here with this envelope virus. And interestingly enough, the envelope is actually made up of phospholipids, the same thing that makes up our cell membrane in our cells. And that's because viruses, if they're meant to be envelope viruses, they will actually take a piece of the host cell membrane with them when they leave a host cell to serve as their envelope. So that's why their envelope is made up of phospholipids, the same as our cell membrane, because they actually take it from the cell membrane of the host cell they infect. And then out here in the envelope, these little blue structures surrounding it, these are known as, they're technically called glycoprotein spikes, but we can just refer to them as spikes. But these spikes here on the surface of the envelope, they are responsible for attaching the virus to a host cell. So I want you to think of those as like keys that are able to attach to locks or receptors on the surface of a host cell. And then if the spikes, which are the keys, match the receptors, which are the locks on the host cell, then the virus will be able to enter the host cell. It is important to know, though, this is an envelope virus, so the spikes are going to be out here on the envelope. If this would be a naked virus, and the virus would not have an envelope, so it would just have a capsid, then those spikes would actually be present there on the capsid. So viruses are going to have spikes regardless, but if they're an enveloped virus, the spikes will be on the envelope. If they are a naked virus and lack an envelope, then the spikes will be there on the capsid.